down. We're going to only talk about physiology at this point. Okay. So I think at this point in the class, which is where I am leaving you for a few weeks and turning you over to Dr. Randler, is really where this class starts to ramp up. And you can tell me if the ramp up was too slow so far. But the material in this chapter, I think, really. So we're talking about primarily chapter 5 today. I think this is material that most of you probably did not see before in previous classes, so we are deep into true physiology now, and there's really a lot to cover. So this is a transition chapter that's very important. So we're primarily focused on membranes today, and this chapter is going to carry you into Dr. Rylander's lectures next week. We're going to be looking at electrochemical properties of membranes. We'll be talking about the Nernst equation, calculating potentials across membranes, and then you're going to move quickly into neurons and muscle and other excitable tissues. In fact, those lectures are already available, and in fact, this lecture is also already posted. So today, our outline, what we're going to cover is we're going to talk about membrane composition first, just some very descriptive properties of membranes. Then we will talk about why are membranes so important anyway? Well, it's one of the reasons is primary reason is they separate components. So I remind you that they separate compartments in the body, and that sets up osmotic equilibrium that we can then look at in some detail. And we'll talk about a case study that highlights the importance of osmotic equilibrium across membranes. Then we'll get into talking about diffusion. So now we're looking at transport, movement of substances across membranes. We'll talk about types of diffusion. We'll look at Fick's law to describe that diffusion, and then we'll talk about other types of transport, which are active transport. And if we have time at the end, we'll also talk about other vesicle-mediated transport processes as well that carry bulk components across or larger components across membranes. So we'll see how we do in time here today. So I really like this detailed view of what's going on inside or at the membrane surface. So you can see a lot of components that are often left out of the other cartoons we're going to show today. So you can see how the cytoskeleton, remember we made a big deal about the internal structure of the cell, links up to the membrane through specific proteins. And you can see the importance of glycoproteins and glycolipids. So carbohydrate molecules are a component that often gets left out of our discussion. We can see how densely this woven fiber of carbohydrates coats the outer side of the membrane. And of course, there's the phospholipid bilayer with a lot of proteins throughout. So just an overview picture. We're going to be talking about a few functions of cell membranes today. Primarily, we're going to focus on the idea that they are separation barriers. They're controlling substances that flow in and out of cells. There are a whole variety of other roles that we could get into. We won't really focus on in this course. We could talk about, later on, you'll certainly talk about membranes as communication devices, as mediators of signaling, in particular from neuron to neuron, transmitting impulses, transmitting chemical signals, transmitting electrical signals. Of course, they also provide some structural support. And this is kind of a high-level, higher scale view of what membranes can do. Looking down at the single cell level, well, there's a lot of specific processes that membranes are involved in. That is, intracellular membranes are involved in things like transporting substances in vesicles across from the Golgi and out of cells through exocytosis, the reverse process bringing substances in through endocytosis. We may get back to these at the end, but I wanted to mention them here just in case we run out of time. Membranes are also really key for steps like the immune system, looking at mediating interactions between invasion of viruses or bacteria, and so it's important in a lot of single cell level processes as well as processes important at the tissue level. So primarily today we're going to be talking about tissue level processes. This is a really cool, I think, technique that lets you zoom in on cell membrane structure. So this is called basically freeze fracture. So what happens, a cell gets kind of, is frozen and very low temperature, and then is kind of cracked in half. And the fracture plane it just happens that it very conveniently follows that hydrophobic interior of the membrane. So you kind of slice right through, cutting the phospholipid membrane in half. In this freeze fracture EM images, you can see if you peel those apart and then look at each side individually, well, they really look very different, right? So you can see proteins embedded on each side of the bilayer, and this sort of increased number of proteins here on the cytoplasmic layer. Here's the extracellular side, so the outside flipped over. A lot more carbohydrates, which you will see in the image here. So this technique has been used to do high resolution structure determination of a variety of different membrane components. And we can classify some of those components in the following way. So the phospholipid particles, fatty acids, cholesterol together, those two components together form the lipid bilayer, which is the sort of core structural feature of those membranes, forming the selective barrier between the cytoplasm and the outside. You can see that these components can also be combined in a variety of other ways. So phospholipids or sphingolipids together with carbohydrates form glycolipids, together with carbohydrates with proteins can form glycoproteins. So all of these components have specialized functions embedded within the cell membrane. Some of those are adding stability, facilitating cell recognition, or cell and foreign component recognition, like through the immune system. Different membranes in the body have different compositions. So that's just interesting, I think, to keep in mind that a red blood cell membrane, we're going to talk today about a lot of general membranes and look at flow materials across them. But throughout the body, the composition of those membranes tunes their biological properties and tunes their permeability to specific substances. We'll come back to that a little bit when we talk about Fick's law. But you can see, for example, that the composition of a red blood cell membrane is very, very different from the myelin membrane that surrounds nerve cells. And that makes sense, but a lot of times when you look at a cartoon in the textbook, it all looks like a lipid bilayer, right? Phospholipid bilayer. And you don't really pay much attention to those subtleties. And again, those inner cell membranes, intracellular cell structures, like the inner mitochondrial membrane that we talked a lot about in the last class, again, have yet a different composition. So, for example, that inner mitochondrial membrane has a lot more protein components, as well as some lipid, but very little, or zero actually, carbohydrates. Carbohydrates in these membranes are generally on the extracellular side. Whereas a red blood cell membrane, you see, has a lot of protein, a lot of lipid, and a myelin membrane is kind of exactly opposite, right? Because those fatty acid components become really critical for transmitting nerve impulses in this specialized membrane. So membranes are composed of substances that are tuned to their particular biological actions. And we can zoom in a little bit more and look at some of those molecules. These are molecules that we talked about in the very first, lecture 2, the first full lecture that you had with me. And you've seen some of these structures before, but I just wanted to remind you how they look. So phospholipid, long hydrophobic chains. When you see that, you should immediately be looking at we talked about saturated versus unsaturated fatty acid being attached to the phosphate backbone of the phospholipid molecules. And we'll see how in a little bit how those saturated versus unsaturated fatty acids change the mechanical and chemical properties of the membranes. And also cholesterol, another important membrane component, It can be inserted into this phospholipid bilayer, and it's kind of, I draw your attention here to the kind of structure of cholesterol. It has a chain as well as these planar ring structure at the end and a polar group, and that's going to insert in a specific way in membranes as well. So those are the lipid components. Let's also just look at this overview schematic of the protein components. And while we maybe have touched on their different structural roles, we talked a lot about integrins and other components. They're protein components that are inserted into the membrane that have specific roles, mediating attachment sites, attachment to other neighbors, attachment to the extracellular matrix, particularly in connective tissue, and epithelial cells where they're mediating cell-cell contacts. We talked about adhesion proteins, cadherins, other proteins that hook up to the cytoskeleton and mediate structural relationships. And here we can focus a little bit more on the functional relationships of those proteins. Today we're going to focus mostly on this category of membrane transporters. We'll be getting back to a variety of different types of membrane transport proteins today. But aside from transport across membranes, remember there are all of these other classes of protein components inserted into membranes carrying out specific functions. So don't forget that there are also those structural proteins as well. Okay, so that's really all I'm going to say about all the different molecular players and the composition. And now I want to get a little bit into what are membranes doing for us. And we're going to focus on this particular aspect, membranes as a way to maintain barriers between the different compartments in the body. So if you read the textbook, it kind of jumps, I feel like it jumps a little bit. The first half of the chapter is pretty different, or the first, say, 1/3 of the chapter is kind of different from the rest. So I wanted to touch on that first. That part talks about exactly what we'll see here, which is that there are different compartments of fluid in the body. So we're taking a real high-level view at this point, and looking at the body level. And then we're going to look at transport of fluid between those compartments. So I wanted to remind you that there are two compartments of fluid, and I think we mentioned them in the last lecture. That's the cells, the intracellular fluid, and the extracellular fluid that, remember, we said surrounds the cells, and that everything that's entering the body from the external environment has to proceed through that extracellular fluid, right? And we can now, today what we'll do is we'll further divide down that extracellular fluid into some subcompartments. So that is the plasma subcompartment and the interstitial fluid. So plasma we saw on the first, lecture 2, we already saw that. That's the liquid matrix component of the blood, right? So remember that blood is one of the four, is part of the connective tissue. We categorize it by the 4 primary tissue types. And we said that's a little bit strange, kind of an aberration, but the plasma represents the matrix. So if you look at blood, we saw a micrograph, you see the blood cells separated widely from each other, right, by this liquid component. So plasma, you know what that is now, and that's of course found within the circulation. Whereas the interstitial fluid is that fluid that lies in between the circulatory system and the cells. Okay, so is everybody clear about all those different compartments I just mentioned? So we're subdividing down a little bit further. And the composition of those compartments and their subcompartments, of course, we said before, it's not the same. They exist in a chemical disequilibrium that's shown in the bottom plot. So the goal of homeostasis, remember in the homeostasis lecture, we said is not to make everything the same everywhere, but to maintain these dynamic disequilibrium states. So we have, for example, wildly different levels of of protein and of a bit of potassium across these different subcompartments in the rock. So the proteins and large anions, those are concentrated in the plasma. They can't cross the blood vessel epithelium, so they don't get into the interstitial fluid. And on the other side, sort of conversely, there's small molecules that can pass through, and they equilibrate into the subcompartments more easily. So between those two, there's differences in ion concentration. There's also differences in electrical disequilibrium, which we're going to get back to with Dr. Reisinger. But one thing that is in equilibrium between the intracellular and the extracellular components, there is osmotic equilibrium, right? And that's fortunate for us. So the fluid concentrations, that means, are equal on each side of the cell membrane. And here we're going to be talking about the amount of solute per unit volume, right? So that means, when I say fortunately, that means that if those are balanced, that means that the osmotic pressures are also usually in normal homeostasis are also balanced. So osmotic pressure here is defined for you. It is very simply the pressure that would need to be applied here on this piston, for example, to oppose the movement of water by through osmosis. So in this example, we placed this selectively permeable membrane here. It allows water, but not glucose molecules, to pass through. And so what's going to happen? Water is going to flow by osmosis into the more concentrated solution, thereby increasing the volume on that side. And the osmotic pressure is very simply that force that would have to be applied to oppose that movement to reset the volumes as shown on the bottom. And so I wanted to mention that in many determinations of chemical concentration that you encounter in the lab, in your careers, it's convenient to use molarity as a measure. That's the number of moles dissolved solute per unit volume, right? And here we're introducing this alternative measure. We have a good reason for that, because the number of moles doesn't include any information about how many particles are contributing to this osmotic pressure. So let's just stop and think about that for just a second. So if you have a glucose solution of 1 molar glucose solution, 1 mole per liter, and its molarity is actually the same as its osmolarity. That means there's 1 particle of that glucose contributing to osmotic pressure. However, if you have a substance that dissociates when you place it in water, so like sodium chloride, right? So take salt, put it into water, there's now 2 particles for each starting NaCl molecule. So there, in this case, actually, we're going to say rather than 2 molecules, we have some incomplete dissociation because we're going to work at physiological conditions. So actually the NaCl won't completely, all of it will not dissociate. So let's say we end up with about 1.8 particles per unit volume. At any rate, we have an osmolarity of 1.8 rather than 1. So that says there are 1.8 particles for each molecule that are contributing to that reaction. So the osmolarity is significantly higher than a glucose solution of the same molarity. Yes?
So I'm kind of confused. The particle for glucose, is that mean basically a particle of water for each particle, like molecule of glucose?
So it means that the glucose molecule doesn't break apart, so it remains as C6H12O6. So 1 molecule will push back. Okay, so just to say that when you see, so this unit is probably a new one for some of you. And also to say that osmolarity doesn't tell you anything about, it doesn't rely on it doesn't tell you anything about the identity of the substance, right? So it only tells you the number of particles that are available to oppose that pressure force. And why is this important in physiology? Well, along with osmolarity, comes tonicity, which is simply the volume change that a cell will experience when it's placed in a solution. And this measurement, you should consider this measurement taking place after, so you take a cell, you place it into this particular solution of interest, and now you let things come to equilibrium. So of course there is going to be, you can always posit there is some time lag there, but when that comes to equilibrium, the question is there a volume change or not? And does the cell expand or decrease in volume? So the same for everyone is contagious. And silence fine. Okay, so let's look here for just a minute. So measures of osmolarity, we just said, we didn't say yet. Sorry, see, this it just confuses me with cell phones. So osmolarity, we're going to compare one solution to each other. So we can say, for example, that A has a higher osmolarity than B. This hypo has a lower osmolarity than B. Now let's look at tonicity. We actually can't predict tonicity from knowing the osmolarity because here the volume change is going to depend on the type of solute particles. So it's not a colligative property. It depends on whether or not those particles are cell permeable. So for example, a solution, when a cell is placed in it, causes the cell to swell, we would say that solution is hypotonic, and if the cell shrinks, the solution is hypertonic. And that's some additional and new terminology that you just really need to be comfortable with so that you can quickly evaluate particular solutions. And there is a problem set question that addresses a little bit of this osmolarity and tonicity balance. But I wanted to point out that there is a critical feature that is not introduced really in the book at this point, but that I think is that you need to know to understand why we have osmotic equilibrium. And that's the question, well, how is the water getting across cell membranes, right? So we haven't gotten too much into the composition of particular proteins in cell membranes yet, but I just told you that water is going to flow in response to being placed in different solute solutions. So how is that happening? Yes, sir.
I have a quick question about the cell growing whenever you have tonicity. What membrane features determine how much the cell can swell before something bad happens?
Yes, so the question is, what features of the membrane determine how much it will swell before something bad happens? And good question. So we are actually going to, in a couple minutes, get to, or maybe 1 or 2 minutes, get to a case where we'll see some of the pathophysiology that can happen. So features that, let's quickly, what features do you guys think will affect how much can a cell change its volume?
Elasticity.
Did you say plasticity or elasticity?
Elasticity.
Elasticity, which will rely on what? What could you change to change the elasticity of the cell membrane? Cholesterol, that's a great one. Protein concentration is another one. So all of those will affect fluidity of the membrane. At the end?
Don't worry about it.
You're just talking to them, okay. Yes.
Would that also depend on the cell type?
The cell type, right? Because I just showed you that table, right? And it pointed out how the composition of membranes, relative proportions of lipid and protein are quite different from one cell type to another. So you could reasonably assume some of them are much more sensitive to swelling compared with others because their protein to lipid ratios are different. Yes?
For swelling, is it only dependent on the cell membrane or does it also be on the size?
Yeah, did you hear what she said? No. Okay, so her question is a great question, and it's does this only depend on the cell membrane, the composition, the relative proportions of different molecules in the cell membrane, or does the cytoplasm matter too? And I'll say yes, the cytoplasm matters too. And let me ask you a question. Do you think that, so at what point, how much do you, well, I don't want to get too quantifiable, let's see. The problems that arise from cells changing their volume, whether it's shrinking or expanding, actually arise long before the cell is in danger of exploding and lysing its contents everywhere, right? So it would, you might think, you have a change in osmotic disequilibrium is set up and cells explode and then you're in trouble. Actually, your troubles begin long before the cells explode because these biochemical reactions happening in the cytoplasm are, the balance is so delicate that they are disrupted and basic cellular processes are disrupted long before the structural integrity of the cell breaks apart. So the cytoplasm is actually really essential in determining your overall response to that hypo or hypertonic situation. Great. Okay, so let's get back to this really critical feature of water. So how does water get across cell membranes? And I ask this because we're talking about a very hydrophobic bilayer, right? So, and I just said, oh, it's flowing, it's going to flow very easily from one side to the other of this ideally selectively permeable membrane. I talked about the water flowing to resist the osmotic differential and the glucose molecules being trapped. But I never said anything about, now we're looking at cells and, well, how is the water going from one side to the other so easily when it's a polar molecule, right? It's a highly polar molecule. And I don't think the book really makes a big deal about it, but I think it's really critical. So does anybody know? It's like very brief. Sorry. Go ahead.
Every cell has like aquaporins.
Yeah, aquaporins. Does everybody know about aquaporins? Everybody knows about aquaporins. All right. So surprisingly, though, people didn't know about aquaporins until 1990s, late 1980s, 1990s. So there was some suspicion that there had to be a water channel because water needed to flow very rapidly, and there probably, so I cheat a little bit. Okay, so water can move slowly through, diffuse on its own slowly through that hydrophobic bilayer, but it's going to take a long time on relative to the scale at which we need chemical reactions to occur. So there was much thinking that there was probably a water channel, and surprisingly, no one knew what that channel protein was until around 1998 or so, when Peter Eger, he was actually studying the Rh proteins in blood, so that's one of the Rh factor, and discovered entirely by serendipity that there was this contaminant protein, was always coming out in their protein preps around 28 kilodaltons. They couldn't get rid of it. They thought it was a piece of an Rh factor protein that was like a byproduct that was getting broken down as they were isolating protein. Instead of ignoring that inconvenient results that kept making their protein preps look really ugly and impure, they actually went and bothered to figure out what it was. And lo and behold, it turned out to be this novel protein that had not been seen or described before. And they went on to look at high resolution to create to crystallize the protein and then to get crystal structures of the protein and to even understand how it worked. And he was awarded the Nobel Prize just in 2003. And I found this awesome animation of an aquaporin, and I feel that you will never forget aquaporins if you see them in this way. That's truly, I don't know if we can do sound, but let's just feel. I don't think we can do sound. It's amazing. Can you hear it? Can you hear it? Okay. That's all. Okay. So the key points are almost all cells have aquaporin proteins, and as such, those channels are what allow water to flow from one cell to another. And we'll have to put the credit up on the website. I don't think you're going to do that. Okay. All right. So super important discovery, and you could really see in that animation what I liked about it is that it's very clear how the water flips when it gets to the inside, and then and how that prevents other components like ions from crossing. So we're going to get to some other examples of membrane transporters later in the lecture, but suffice to say this is a true channel protein in that it's open to both sides of the cells simultaneously. There's questions.
Does it ever close? Like if the cell doesn't want to water like plants?
Does it close? Yeah, I think that it is able to open up and accept more versus close. And I do think that is true. Yeah. And that is part of the regulation. Another thought about aquaporins, but let's go ahead and look at this case study. And maybe I'll look at it as we're talking through this. Okay, so you are part of the craft team of quantitative engineers, and today you're observing in the emergency room of the local hospital. While you are there, a 21-year-old woman is brought in. She's supported into the ER by two of her friends. She is not able to speak. She appears confused, a bit delirious, clearly weak muscles, fatigue, loss of energy, balance, and so on. So she's not able to answer your questions. But while the medical team goes on and attends to the patient and orders some tests, which we'll see the results of in a minute, you and your team start to talk to her friend. You know what happened. Of course, they are very upset. And upon discussion with them, they reveal to you that they had been with the woman the night before. They were at a party. Perhaps what happened was that she ingested some of this substance, methylene dimethamphetamine, MDMA, also known as ecstasy. So she was at a party, she took this drug. Question over here? Okay. You inquire for, so this is a synthetic psychoactive drug, chemically similar to the stimulant methamphetamine. It has both stimulant and psychedelic properties. And you ask about, do they know if she took any other, was she on any medications? They don't think so. The friends say no, probably not. Any other substances that she could have taken that would have interacted? Was she drinking alcohol? They say no. Actually, she didn't want to drink. She might have had a beer, but she didn't really want to drink any alcohol because she had a test to study for. She was really worried that she didn't want to. Hopefully this never happens to her. She didn't want to have a hangover the next day, so she was drinking a lot of water, and she had eaten dinner before the party. She wanted to spend the next day studying for an exam. Okay, so in the meantime, the medical team has come back with their, they have ordered a blood panel, and here is the blood panel. So I'll give you a minute to look at this slide. So the normal range is shown there versus the patient's readings. And I want to remind you, this is the blood, so this is primarily plasma levels of these different components. So when you think about which compartment of the body is affected here, you're looking at plasma readings. So after you have sort of scanned it, to give you a minute to scan through. So what is primarily affected? What do you think is
Sodium, potassium.
ion? They're salt ions.
So can I jump forward? Or,
You want to jump forward?
like, I think what
Want to come up here?
electrolytes, like my
What do you
theory
think
is
is
that
happening?
the electrolytes are off balance, so her nervous system is completely thrown off. That's the delirium.
Okay, so her electrolytes are clearly, basically they are low here, right? So let's see what's low. Potassium is low, chloride is low, sodium is low. And you think that is probably the nervous system effect is a result of low levels of electrolytes, you have to say. And, okay, well certainly the salt concentrations are low here. And let's look at then her cells. Should we look at it? So before I turn to the next slide, why don't you tell me, what do you think about the concentrations of these ions, sodium, potassium, chloride? How do you think they look inside her cells versus outside her cells? So which is higher? There's orange shirt. All right, red shirt on the side.
I think they're higher inside the cell. And
Yes.
so then the water would want to pass into cells in order to balance out.
Great. Okay, so he says, so remember these are the plasma levels. So in the plasma, there is lower than normal salt concentrations. And he says, well, inside the cells, that means that there is going to be higher than, that the concentration of those salt ions is going to be higher compared with outside the cells. Does everybody agree? Yes. Okay, great. Does anybody disagree? No. We all feel good about this. Okay. And because of aquaporin in the cell membrane, water is going to move, right? And so actually he jumped to anticipated my next question. So water is going to move. We have inside the cells a bit higher concentrations of salt than outside of the cells. And you think water is going to move inside the cells, you said, right? And okay, so actually sometimes I give you these multiple choice in class because I think you probably know that the exams are going to be multiple choice in this. You may have heard rumors of that from last year's classes. So in general, except for a few identification questions that Dr. Mauser also shows when he does histology, most of the exam questions are multiple choice. So you already sort of narrowed it down for us quite a bit. Very helpful. So which answer do you think is the most correct here? C. Is this a thing that we do this? It's awesome. I like that. Okay. You can learn to do signing answers. Yes. Okay. So water is going to move. Water is moving in both directions because aquaporin is everywhere, right? And aquaporin can transport that water and allow it to move through. But the net flow is going to be inside cells. Great. Okay. Good. So this is a condition called hyponatremia. And what is the problem? So essentially, the patient, remember I said was, she took this ecstasy. She was also drinking some water throughout the evening. And so normally your kidneys would respond to, you drink a lot of water, would respond by producing large amounts of dilute urine. But however, ecstasy, one of the other effects of the compound is to act as an antidiuretic. We're not going to talk about kidney today, but suffice to say that it instead forces the kidneys to make more concentrated urine. So overall, what's happening is the patient has excess water, right, building up inside of cells, and her electrolyte levels in the plasma are falling, as you mentioned. And so there is osmotic disequilibrium happening here. So there's acute swelling of cells and tissues as a response. And this is especially problematic in the brain. Why do you think it's especially a problem in the brain? Neuron fibers.
For your brain, right?
Yeah, Courtney says there's just not enough space in there for your tissue to expand. So anytime your brain is swelling, it's problematic because there's just nowhere for that tissue to expand into, right? I'm surrounded by a skull. So this can very rapidly become a serious and potentially fatal situation. So the treatment is to administer IV fluid, possibly with a little bit higher than normal sodium concentrations to quickly correct that salt imbalance. And with rapid treatment, this patient recovered, so let's have a happy ending for her. But unfortunately, there are similar cases of, remember this word, hyponatremia, that have resulted in death. And so you see, not just, so ecstasy can be a side effect of drugs, also instances where people just drink, the silly radio contest, terrible and tragic as well. Just drink, people who are drinking too much water and for whatever reason have a toxic buildup of water that resets their osmotic pressure, moves it into disequilibrium. So occasionally you hear about long distance runners who are consuming only water without replacing electrolytes as well, and this is very rare. You are far more likely to succumb to dehydration when you are exercising than to go into hyponatremia. I know when I was doing a lot of running, my parents would read these articles in the newspaper and then call me and say, drink less water. Well, so this is rare. It can happen, but it's not happening that commonly. Of course, if you have some kidney issue, perhaps occult issue that's not diagnosed, that can complicate things. Yes, sir?
Did the ecstasy have anything to do with this?
Yeah, so actually the ecstasy did act as an antidiuretic. So was part of the reason why there was, the kidneys were not able to keep up with the removal of water from the plasma. Yeah. So that was part of it. We won't talk about kidney function here, so we won't get into the details of how the kidney was impaired, but that is part of it. So allegedly, Andy Warhol, the famous artist, he went into the hospital for gallbladder surgery and was given, I say allegedly because it was settled out of court and we don't really know, but the records indicate that perhaps one of the reasons he died. So he went through the surgery just fine, went to post-op care, and then over that night was given IV fluids and perhaps was given too much IV fluids as a medical error because he was quite underweight and he already had some complicating issues, and maybe they didn't calculate appropriately the amount of IV that he should have received post-surgery. And as well, another case where this comes up is particularly in poverty-stricken parts of the world, third world, where infants are given, say, formula is flown in by a relief organization, and that's powdered and is intended to be mixed at appropriate concentration, and instead people are kind of rationing it, trying to get it to last longer, and is therefore mixed up inappropriately at a more dilute concentration. Well, if that is the sole source of the infant's fluid as well as nutrition, that can become also problematic in that they're taking in too much water, especially if there's complicated medical issues there. So these are all cases, again, it's a rare condition, but here's where I also wanted to mention that all of these problems come on pretty rapidly. You can see they set on in the hours after this osmotic disequilibrium was created, and they didn't need the cells to lyse. So no cells needed to get to the point where they exploded in order for there to be some really severe pathologies here. Okay, so I think there we will end our discussion of osmosis itself, and we're going to move on to talk about diffusion and other types of membrane transport. So here's kind of an overall schematic that you can find in your textbook of the different ways that substances can go across membranes. So depending on, first here, this division of whether the process requires energy or not. And we'll go through each of these four types in some detail. If we have time at the end, which I suspect we may not, we will try to get to this family of processes, which is dealing with much larger particles or the internalization of like a bulk amount of fluid at once. Those of you who maybe, if you work in labs in the department where we have some of the best researchers studying nanoparticles in this department, these are processes that are important for following the uptake of those particles into cells. Okay, let's go back here. So, but what is in common? These processes are talking about much larger substances. Over here, in the most of the remaining time, we're going to talk about objects that are many times smaller than the ones dealt with over here. So we're going to talk about ions and small molecules moving across membranes. Okay, so we're over here at diffusion. So some basic properties of diffusion. First of all, is a passive process. Molecules are going to go from high to low concentration, so down their concentration gradient, and there will be a net movement of molecules proceeds until dynamic equilibrium is reached. So the concentration will be the same then everywhere. And of course that means, I just remind you from a couple lectures ago, that individual molecules are still moving at equilibrium. It's just, of course, that for each molecule that exits, another one is entering. Diffusion occurs rapidly over short distances, slower over long distances. It's directly related to temperature, which is important because these properties hold for all biological membranes, but maybe in cells we cannot tune the temperature too much. However, if you are reconstituting some of these systems in a tube, you might want to know that your temperature is in the appropriate range because that will affect the rate of diffusion. And as well, it's inversely related to molecular weight and size. So smaller molecules, I think it's intuitive that they require less energy to move over a distance and therefore are likely to diffuse faster. And just a note that we can talk about diffusion in an open system, or we can talk about it across compartments, which is primarily what we'll be talking about in this class. And also another kind of special note that when we think about ions, those have to always move in response to their concentration gradient, but also their electrical gradient. So we're going to talk more about ions in the excitable cells in the next week, but just wanted to make a note there that there's a couple of things to think about when you think about charge particles. So simple diffusion, passive transport across a cell membrane. Here we're talking primarily about small, non-polar molecules, readily soluble in lipids, and they can be transported this way. So of these choices, which one do you think would be one that could move by simple diffusion across the lipid bilayer? D, yeah, this now. Okay, so D, oxygen, it is small, it is non-polar. So how come, what about on your skin? Is oxygen diffusing by simple diffusion across my skin epithelium? Remember, that's epithelial cells in the skin. Do you think so?
No.
No, why not? We're not what? What is it?
We're not frogs.
Frogs. We're not frogs. And what distinguishes us from frogs?
You don't breathe through our skin.
Right, okay. So why? Why are my skin cells not diffusing? What is preventing the oxygen from going? They're dead, yeah. So there's a layer of I remember we talked about extracellular matrix protein, keratin, and that is blocking the diffusion of oxygen into those skin epithelium. And as well, really for the oxygen to be transported across by simple diffusion, there should be some water, so it should be happening in aqueous. So neither of those things is present there. Okay, but yes, oxygen does easily diffuse by simple
Does it
diffusion.
always have to be aqueous?
Pretty much for what we're talking about, we're always going to deal with an aqueous situation. But certainly, in a general sense, we could talk about diffusion of gases from this side of the room to that, right? Okay, so what affects the rate of diffusion across a membrane? So here, we introduced Fick's law, which is this describes this relationship. So diffusion rate increases when the surface area, the concentration gradient, or the membrane permeability increase. It's related to all of those. Out of these factors, I think the most complicated one to really think about is membrane permeability. That can encompass quite a lot of different factors. There are a lot of things that contribute to membrane permeability. That includes the size of the diffusing molecules, the shape of those diffusing molecules, the lipid solubility of those molecules in the bilayer. And we already mentioned a little bit, one of the questions touched on the composition of that lipid bilayer. So for example, adding different protein components will change permeability of that, of the lipid layer. And okay, so here's our lipid bilayer. There's lateral movement within it. There's occasional flip-flopping, but very rare across it. But there's definitely lateral movement. What are some of the factors that affect fluidity of that membrane? We started to say this, but I wanted to show the image as well. We talked about saturated and unsaturated fatty acids. Those structures behave very differently when they're packed into a membrane structure. So these unsaturated hydrocarbon tails, where they're kind of bent. This is the position of the double bonds, so there's still room for additional hydrogens and oxygens to be added there. They're interacting with each other, those tails, and because of those chemical interactions, they are prevented from forming a close packed structure. So there is significant empty space here that increases the fluidity of that membrane. Right? Whereas here you can see if we have all entirely saturated hydrocarbon chains, and they are packed very close together with very little space. The membrane is much less fluid. And if we take this membrane and then insert cholesterol molecules, so those are going to be sort of spotted throughout the membrane. And here are the cholesterol molecules, which are, remember, these bent kind of planar structures, the rings we insert into the space between the phospholipid hydrocarbon tails. They really reduce phospholipid movement and also disrupt that regular packing structure. So we're not really talking here about cholesterol in the arteries or anything like that. Those are just, those are much more esterified cholesterol molecules. Here we're just talking about the normal role of cholesterol as a component of cell membranes. So it affects the fluidity and by extension affects the permeability. And how do we know that proteins can, I said that there's a lot of lateral movement. Well, how do we know that? Turns out that there are some easy ways to study that. You can take one cell type and another cell type and fluorescently label some particular membrane protein of interest, and then you can create hybrid cells. You can convince the cells to form hybridoma structures, and when they do that, you can note the time that it takes for those fluorescent molecules to redistribute themselves in the membrane of the new cell. And heterokaryons. Hybridoma is a different cell. But at any rate, the mixed proteins can be observed pretty rapidly. They become distributed homogeneously throughout the cell. So there can be pretty rapid lateral diffusion of proteins within a membrane. Remember, we're talking about across a membrane. And so we were here, we can take this relationship and fixed law and rearrange it as in this way, so that now we're looking at diffusion rate per unit of surface area. That's a term that describes flux. So this is flux of a molecule across the membrane, and that you can see now very easily that there's a concentration gradient as well as membrane permeability. Okay, so let's go back to our schematic map, and we were just talking about simple diffusion. Let's talk about facilitated diffusion. Okay, so facilitated diffusion is, remember, still in the arena of processes that do not require the input of energy. So it's a passive process, and it applies to solute molecules like that are polar, glucose, amino acids, or some of these. So what happens? It requires carrier proteins to move the molecule down a concentration gradient. So again, there's no energy input. It always proceeds down the concentration gradient, and because of that, the net movement of the molecules will stop when equilibrium is reached. So there's, imagine a lot of different types of these specialized membrane protein carriers that regulate this process. And some of those are shown here. So there are two main groups of membrane transport proteins. One is channel proteins that span the entire membrane, connecting the two compartments. So its essential space can be selectively opened, permitting transfer of specific molecules that don't typically stop and bind inside the channel, so they pass through. And we saw aquaporin is a great example of one of those channel proteins. So these typically enable transport of water-soluble, polar molecules, ions, molecules in that category. The second main classification of membrane transport proteins are carrier proteins that bind that binds the biomolecule. They don't ever form an open channel in which they're open to both sides at the same time, but they bind, they change conformation, and then they release on one side or another. So there's some conformational change here. And the example that we'll talk about in this category is the GLUT carrier protein, which accomplishes facilitated diffusion of glucose. So this isn't the first time you've heard of this protein, actually in this class, because when we talked about cell respiration and glucose transport in the body, we also mentioned glucose transporters. So here, there's no ATP input, there's no energy required. This protein is going to move glucose down its concentration gradient. It's going to reach equilibrium when the concentration inside and outside are equal. So starting with low concentration inside the cells, the GLUT membrane protein will facilitate crossing of glucose across the cell membrane. And cells actually have a little trick to keep this process going, so they can prevent equilibrium from ever being reached. And they do that by, once the glucose comes into the cell, it is available to the enzyme that converts it to glucose 6-phosphate, which, if you remember from the last lecture, is the first step in the glycolysis pathway. So what happens now? So glycolysis, it proceeds, and that molecule gets channeled through all the enzymatic reactions of glycolysis. But adding that glucose to turn into glucose 6-phosphate keeps the concentration of glucose in the cell low. So effectively, as soon as it comes in, it gets passed along. And that chemical modification also prevents it from being into a transporter and to take it back out of the cell. So it's really maintains that disequilibrium and successfully continues to facilitate diffusion of glucose. Okay, so let's move in the last 5 minutes to the types of membrane transport now that require energy input from ATP. So here we're talking about active transport. And we're talking about 2 types, so primary active transport as well as secondary active transport. We're going to see in this next 5 minutes an example of each one. So active transport, very simply, is moving molecules against their concentration gradient. So sometimes these cells have to move molecules in such a way that they can maintain internal concentrations of solids that are different from concentrations in the environment. And so they need to work